Hello everyone, my name is Diego Marescotti and I'm responsible for the in vitro pharmacology laboratory at Philip Morris International. I would like to start my presentation by thanking the organizer for this opportunity to share with you how we at PMI plan to develop and apply the adverse outcome pathway, abbreviated as AOP, in the context of human health risk assessment. The assessment of human health risk is a multi-step process meant to estimate the nature and probability of adverse health effects in humans exposed to chemical. The first step, the hazard characterization, is often associated with extensive in vivo toxicity testing using standardized and internationally recognized guidelines or protocol. However, considerable time, cost, and animals are required for generating relevant hazard data. For ethical reasons, in vivo testing should always be challenged and be considered only if available in vitro tests are either not applicable or not adequate. Furthermore, the classical in vivo testing approach is based on apical endpoint, which provide minimal information regarding the mode of action and is characterized by several limitations in predicting toxicity to human due to intrinsic interspecies differences. Given this limitation, we have experienced increasing scientific, social, economic, practical, and regulatory pressure within the last decade, which has resulted in the development and use of more mechanistically based alternative chemical testing in the context of human health risk assessment. To this purpose, regulatory frameworks are moving toward risk assessment approaches that better protect human health and are independent of animal testing. Therefore, 21st century scientific research incorporate the use of human-relevant methods that are ethical, scientifically sound, and can accurately predict chemical toxicity. In this context, the scientists at PMI initiated a project to develop and implement new approach methodologies in line with the goals of the 21st century toxicology program. In particular, we choose to utilize the AOP framework launched by the Organization of Economic Cooperation and Development, also known as OECD. The program's activity aims to collect, organize, and evaluate biological information that links upstream molecular alteration to downstream organ, organism, and population changes. An AOP is a structured representation of various biological events that lead to an adverse effect considered relevant to chemical assessment. The AOP links the existing biological knowledge regarding a series of causally connected key events, KEYs, between 2 points, a molecular initiating event, MIE, and an adverse outcome, AO. Moreover, AOPs are deemed relevant for expanding the use of mechanistic toxicological data for risk assessment and regulatory application. Our project has three main objectives. First, we aim to develop AOP-based approach for integration into our product assessment strategy for smoke-free products. Second, we intend to develop predictive computational model to improve the prediction of AO based on preclinical data. Lastly, we seek to engage with partners and regulatory authorities to accomplish the project mission in a collaborative manner. To provide a practical case, in the following section, I will share an example of our approach for deriving in vitro model from an AOP for decreased lung function. Given that selected AOP describe various mechanisms underlying reduced lung function through the impairment of mucociliary clearance, or also known as MCC, let's first have a look at the mechanism in more details. MCC is a self-clearing mechanism of the airway, an innate defense mechanism that protects the pulmonary system from the harmful effect of inhaled particles. MCC-based lung protection relies on two main functional components: the airway surface liquid, ASL, and the cilia on the apical side of ciliated cells. The ESL is a thin layer of fluid that covers the apical surface of the airway epithelium and plays a key role in airway homeostasis. Small particles such as dust, particulate pollutant, and allergens are trapped in the mucous layer, which is continually swept toward the oropharynx by the beating movement of the cilia. Various well-known cellular components such as CFTR and FOXJ1 protein, which play a pivotal role in maintaining functional cilia motion, are essential for efficient MCC. MCC is highly efficient in healthy individuals. However, impairment of any functional component can in turn disturb MCC, and mucus buildup in the airway leads to airflow obstruction, thereby reducing lung function. Such causal relationships are depicted in the AOP selected to establish our first test case. In particular, as illustrated in the flowchart summarizing the AOP network, mechanisms that link oxidant exposure with decreased lung function involving parallel routes and multiple keys have been described. Overall, the selected AOP link increase oxidative stress to decrease CBF and decrease MCC. Our in vitro approach envisioned the use of organotypic bronchial epithelial tissues, as this cellular model exhibits all required phenotypic features, including functional MCC. The first step will be the identification of various assays that match the whole set of endpoints derived from AOP event. Next, all identified assays will be developed using the selected test system and specific reference compound relevant to each endpoint. Once all assays covering at least one AOP are available, we plan to assess the causality of the event using a set of reference stressors. Then we will use the data obtained to train the computational model. Additionally, we will implement a quantitative AOP approach to predict the likelihood of the AO. Let me conclude by acknowledging the contribution of all participating colleagues and current partners working closely with us to develop and establish the project. Lastly, thank you very much for your attention.